Component 1 task book, and this is question 4. And this would be about photosynthesis. You should recognize this diagram here and identify regions A and B. So just straight knowledge, A01, what's A and what's B. You should know those names. Likewise, two pigments that you'd expect to find in A. This is going to be different to B. So you'd expect one in B and then some other suggestions for A. And exactly where you would expect to find these photosystems. So we want you to be pretty exact here. So you're going to narrow it down to kind of the fine detail. So you've got a leaf, and then you've got something in the leaf, and you've got something else within, the something else. So give us the actual place that you know from your notes. Again, A01, recalling knowledge there. Now we move on to A02 stuff, where we've got an experiment. And what's going on here? We're shining light through a filter of a certain colour, and so only a certain colour of light will hit this apparatus inside. We've got a plant inside, and this would be in some liquid pondweed. So pondweed you'd expect in a pond, so this has got liquid inside it, and we are measuring something. We are measuring our oxygen concentration. So our oxygen concentration is going to be given to us by this computer, so we can shine coloured light and see how the oxygen level changes. And think about your photosynthesis equation. What's oxygen in that? Explain how the use of the dissolved electrode allowed the rate of photosynthesis to be determined. So if oxygen changes, what's that telling you? And just relating back to your photosynthesis equation. Give two sources of inaccuracy, and we're thinking, what else is going to be changing in here? So think about, controlled variables. They're usually a source of inaccuracy. So as this experiment progresses, what else is being used up or produced, that sort of thing. What else could be changed that you want to keep the same? So there's one other kind of big obvious thing that you usually keep the same in all these sorts of experiments. Over the page, here's what you would get, or what you are expecting. Wavelength on the bottom, well, that is the same as colour, and in fact we can see there blue and red. The name of the graph, there's two graphs that look very similar, so you just need to pick the right name. The clue is this is the rate of photosynthesis. So the other graph is the rate of absorption of the light, or how the light is absorbed. This is the rate of photosynthesis. So something's actually happening here. So there's a clue. And you just need to draw on the line there, and you got 2 marks for that. So there's 2 things that you need to do, and that's 13 marks.